Hello, everyone. In this chapter, we're going to be looking at the plasma membrane, its structure and function. So in this image, what you're seeing here, these are two different cells. So you're seeing the plasma membrane here, and then the proteins that are located within the membrane are these green, weird finger-like structures. And then these here are vesicles. And so what you're seeing here, these are two neuron cells, and they're sending a signal. And so they'll send the signal containing the signal molecules, and the vesicles will merge with uh, the plasma membrane and release the signal proteins so that they can be transported to the next neuron that will be collected by these signaling proteins in their plasma membrane. So the plasma membrane is super important because one of the things that it controls is cell-to-cell communication. Plasma membranes regulate inbound and outbound traffic. Some small molecules move across the cell membrane using passive transport. In other words, they don't need any energy to go across the membrane. And some require transport proteins or energy to be transported across the membrane. Some small molecules use active transport, which requires both energy and a transport protein. And then if you're very large items, large molecules can move in and out using bulk transport, which is called exo or endocytosis. And so in this chapter, we're going to review all of these um, regulatory methods and ways that things can travel across the membrane, which includes passive transport, active transport, and bulk transport. Cellular membranes are fluid. They are mosaic models of lipids and proteins. Lipids and proteins are the main components of the membrane, but carbohydrates are also very important. They like to attach themselves to either the lipids or the proteins, and they help aid in different kinds of signaling that is necessary from cell to cell. The phospholipids, remember, naturally as they are as molecules, they form a bilayer naturally in nature, and they form a bilayer naturally when it comes to making the plasma membrane. Phospholipids are amphipathic. Amphipathic molecules have a hydrophobic and a hydrophilic region that makes up their bodies. Because phospholipids are amphipathic, they form naturally um, layers with each other. Phospholipids will form a bilayer with the hydrophobic tails inside the membrane, and the hydrophilic heads will be exposed to the water on either side. Most membrane proteins are also amphipathic. Not all proteins that are associated with the membrane are amphipathic. There are sometimes proteins that like to sit on either side of the membrane. They don't go through the membrane. But if you are a protein and you're sitting in the membrane, then you are also amphipathic. There's hydrophilic regions of the protein that are oriented either towards the cytosol, or they're oriented towards the extracellular matrix on the outside of the membrane. And then the hydrophobic regions are the regions that are in between the two bilayers. So in this diagram, you can see this is the bilayer of a plasma membrane, and it's showing you that if you focus on the phospholipids themselves, you can see that the hydrophilic heads are oriented towards the fluid side where water molecules and other liquids will be located. And the hydrophobic tails are going to be oriented towards each other where there's no fluid located. And so these compact phospholipids will prevent um, ions and molecules from traveling through the bilayer, unless it's something that's able to go through the bilayer. Now, the plasma membrane is considered a fluid mosaic model because its membrane structure depicts the membrane as a mosaic of protein molecules bobbing in a fluid bilayer of phospholipids. Proteins are not randomly distributed in the membrane. They often form groups and carry out common functions. However, they're still able to move around and travel within the membrane. So the plasma membrane, in this, every sense of the word, is very fluid in that it's not static, it's not cemented together. The molecules are able to move. However, that bilayer of phospholipids prevents things. It makes the bilayer selective. And so in this image here, you can see all of the components that form the fluid mosaic model. In that there are proteins, big, huge proteins that will travel through the plasma membrane. There are some things that will sit on top, like the integrins here. The peripheral proteins can sit on either side. And so the extracellular matrix scaffolds the outside of the cell, and then the microfilaments of the cytoskeleton will scaffold the inside of the cell. So the membrane is fluid. Membranes are held together by the weak hydrophobic interactions um, of the phospholipids. Most of the lipids and some proteins can move sideways within the membrane. Rarely, a phospholipid will flip flop across the membrane from one phospholipid layer to the other. So, as temperatures cool, membranes will switch from a fluid state to a solid state. The temperature at which a membrane solidifies depends on the types of lipids that are found in that plasma membrane. Membranes that are rich in unsaturated fatty acids are more fluid than those in saturated fatty acids. Remember, we covered saturated and unsaturated um, lipids in chapter 5. So membranes must be fluid to work properly. So unsaturated versus saturated. If it's unsaturated, it's more fluid. If it's viscous, then it's made of saturated phospholipids. So depending on you, of course, your membranes can be made of a variety of both things. Cholesterol is a membrane component in animal cells that is of high importance because it has variable effects on the fluidity of the membrane. Now, remember when we covered cholesterol in chapter 5, I talked about how cholesterol could be a bad thing because the builds up in your the building up of cholesterol can harden your arteries, leading to, a form of heart disease. But cholesterol is important in the fluidity of membranes. At warm temperatures, such as 37 degrees Celsius, cholesterol restrains the movement of the phospholipids. And then at cool temperatures, it maintains fluidity by preventing tight packing. It's a component in the phospholipid bilayer, and so it creates space in the bilayer. Now, the question is, why does it need to prevent or restrain movement of phospholipids when it's warm? Because movement of molecules, which we'll learn in chapter 8, creates heat. And so we don't want to keep getting hotter, we want to get cooler. At cool temperatures, we want to stay fluid. We don't want to get packing too tight because then we would freeze. So cholesterol has a very important job in maintaining fluidity of your cells. Plants use different but related steroid lipids to buffer membrane fluidity. They don't use cholesterol, they use other steroid lipids that are similar in many ways. So, cholesterol within an animal cell membrane reduces the membrane fluidity at moderate temperatures, but reducing phospholipid movement, and it hinders solidification at low temperatures by disrupting the packing. So these three examples here, the either unsaturated or saturated hydrocarbon tails in the phospholipids or cholesterol are all used to maintain the fluid nature of the phospholipid bilayer. So membranes must be fluid to work properly. Fluidity affects both permeability and movement of transport proteins. If you need to ship your transport proteins to a different side of the cell, it must be able to move. Membranes that are too fluid cannot support protein function. So there is a balance that must be made in the cell in the fluidity of its membrane. And as we saw with the beginning, the neuron cells, being able to function and have a very quick reaction is important for biological function. So, organisms that live in extreme temperatures have adaptive differences in membrane lipid composition. They have adapted to different environmental situations. And so the evolution of the differences in membrane lipid composition allows for certain organisms to survive in different habitats. Um, for example, cell membranes have a high proportion of unsaturated hydrocarbon tails in fish that live in extreme cold temperatures, which makes sense. They live in the cold, so having more unsaturated um, phospholipids is more important compared to something that lives in a hotter, warmer temperature. Some organisms are able to change their lipid composition in response to changing temperature. For example, in winter wheat, the percentage of unsaturated phospholipids increases in autumn to prevent the membrane solidification during winter. So it prevents it from being able to be frozen. Um, the phospholipid bilayer is also composed of the membrane proteins, and then their functions are super important. So a membrane is a collage of different proteins that are often cluster in groups because they're searching for signals. They're embedded in the fluid matrix. So this structure resembles a tile mosaic. And I threw this image in here because I wanted you to show that this is a phospholipid bilayer. You have your green phospholipids, and then you have proteins also, which are red and blue. It creates a mosaic, a variety of huge form. It's very diverse of components. And the protein composition of the membrane can vary within an organism between the cells, the different types of cells. It's all very, very different, and it can be selected for whenever you're trying to, when a cell is becoming a certain type. There are two major types of membrane proteins. There are peripheral proteins. These are proteins that will bind to the surface of the plasma membrane. They don't actually go through the membrane, but they will bind to the surface of the membrane. Now, the plasma membrane is this huge structure that surrounds the cell. And so it creates all this space, and cells need a lot of space to do all of the chemical reactions that they need to do. And so cells will often utilize the space with peripheral proteins. There are integral proteins, meaning they integrate into the plasma membrane, so they penetrate the hydrophobic core. And then, of course, if it's an integral protein, then it's usually a transmembrane protein, which are integral proteins that span the membrane, meaning that they have both hydrophobic and hydrophilic regions of the protein. And so in this example, it's showing you that this is a transmembrane protein that's going all the way through the plasma membrane, which contains hydrophobic and hydrophilic regions. The hydrophobic regions here are alpha helices, and then the ends of the protein, which are labeled as the N terminus, the C terminus, are the different sites, the binding areas that exist on those proteins. Some proteins are held in place by the attachment to the cytoskeleton on the inside of the cell, and then some are held together by attachment to the extracellular matrix on the outside of the cell. For example, proteins called integrins will attach to the fibers on the extracellular matrix. So the cell surface membrane proteins can carry out several functions, including transport, enzymatic activity, signal transduction, cell-to-cell communication or recognition, intercellular joining, and then attachments to the ECM. So in this image, you can see there's examples of all of those. For example, transport. There are transport proteins, which we're going to cover very much in depth, that allow for certain solutes to travel through the membrane. It can either be passive or a form of active transport, and we're going to cover that in the next the next video. Enzymes can sit in the membrane, like for example, they're just using the membrane as a space to do um, as an area where they can do chemical reactions. Enzymes are highly important in that they allow for chemical reactions to occur. They're catalysts, and we'll cover them more in depth as well. Signal molecules. So we have signal transduction in that we have proteins that are labeled as receptors, and they are able to obtain signals in the form of either a protein or a solute that will bind to the receptor, and it will send a signal towards the nucleus and let the nucleus know, okay, this is happening, and then it will trigger a chain reaction where the nucleus will create a pathway for something to occur. For it could be the creation of um, a protein or something that needs to be done by the cell. There are proteins that are responsible for cell to cell communication and recognition. And so we have usually glycoproteins. It's a protein with a carbohydrate attached to it. And so, for example, you will have cells. The example is your white blood cells that will travel through your body. So you have white blood cells that are traveling through the body, and they're searching for invaders. And how do they know that you are not an invader? It's because you usually have a um, a fingerprint protein that's this glycoprotein that lets it know, okay, hey, I'm actually part of the body. There's intercellular joining. This is where cells can bind together. For example, this is also important in like the junctions, tight junctions, for example, where you can form and fuse, not fuse, uh, bind the two cells together by their intercellular joining proteins. Then, of course, there's attachments to the extracellular matrix, and so there's like the integrin peripheral proteins that it can attach to the fibers of the extracellular matrix. The cell surface proteins are important in medicine. For example, the glycoproteins or fingerprint proteins. For example, HIV can enter an immune cell by binding to a cell surface protein called CD4 and then a co-receptor CCR5. Um, so it's been discovered that individuals lacking CCR5 are immune to HIV. And so there are drugs that are in development to mask CCR5 and block HIV from entering the cell. And so in this image, you can see in a normal situation, in an individual that still has the co-receptor, HIV will attach to the receptor on the membrane. And then so attaching to the receptor means it's now able to fuse its viral envelope with the plasma membrane of the cell. And so the HIV can then enter into the cell. And then the individual's then infected with HIV. But then if you are missing your CCR5, HIV has no way of entering the cell. So it's fascinating that this is a process that has been discovered. And as many of you might know, um, the ability to treat HIV is bordering on the curable side now than on the treatment side. So in the next, I would say 10 years for sure, HIV would probably will um, the drugs will be available for the prevention of infection or curing of the disease in general. So the role of membrane carbohydrates in cell-to-cell recognition is important. Cells recognize each other by binding to the molecules on the surface of the membrane. Many of these surface molecules are bound to short branch chains of carbohydrates. So we gave an example of like the glycoproteins. Those are proteins that have carbohydrates attached to them. However, the carbohydrates can attach to the lipids as well, and we call those glycolipids. And so, depending on the the protein and the carbohydrate, they can do different things. They can be signals for different things, but their diversity of um, the potential diversity of the mixtures of proteins and carbs can enable them to function as markers for cell identification. Uh, the the bilayer has a distinct um, sidedness, and so uh, the synthesis and sidedness of membranes is actually important. Membranes have distinct inside and outside faces. The inside, which is facing towards um, the cytosol and cytoplasm, is very different than the one that's on the outside facing the extracellular matrix. The composition and distribution of the proteins, lipids, and carbohydrates is asymmetrical across the membrane, meaning that one side is not the same as the other side of the bilayer. Um, and that can generally be true for both the plasma membrane and any membrane structure that's inside of the cell, from ranging from the ER to the Golgi apparatus. The sidedness, the different sides of the bilayer can be different. In the next video, we're going to cover, well, you know what, guys? I'm going to include this section in, because it's very short in this video. And then the next video, we're going to cover diffusion. So before we get to diffusion, let's do membrane structure and how it's selectively permeable. So the plasma membrane controls the exchange of materials between the cell and its environments. The plasma membrane is selectively permeable. It has selective permeability. So some substances can cross across the membrane very easily, and some cannot. And so the fluid mosaic model explains how membranes regulate molecules across the membrane. And so I like to explain it. It's kind of like a screened window. The screen in the window. So the reason why we have screens in windows is because we want to keep bugs and little critters out if we open our windows. So we want to be able to open our window and keep things out, but we want to open our windows so that we can let air in, right? So screens, protective screens for windows or doors are selective membranes. They are selective, they have selective permeability. They only allow certain things to go through to come in, and some things are not allowed to come into there. It prevents it from coming in. So the lipid bilayer, it acts the exact same way. Hydrophobic molecules dissolve in the lipid bilayer and pass through the membrane very easily and rapidly. For example, hydrocarbons or carbon dioxide, oxygen gas will pass through the membrane very easily. There's nothing that can stop it to go through. The hydrophobic interior of the membrane, though, will prevent hydrophilic molecules from traveling through the membrane, which is usually much larger items like sugars, water, and other ions. If they pass through, it's very slowly, if at all. So there have to be transport proteins to allow for things to go through the membrane if it can't easily go through the plasma membrane on its own. So hydrophilic substances can cross through using transport proteins. Transport proteins can be either channel proteins or carrier proteins. Channel proteins have a hydrophilic channel that certain molecules or ions can use as a tunnel. Carrier proteins will bind to the molecule and change shape to like release them on the other side of the membrane. So channel proteins, the solutes can just flow through. Carrier proteins, the item will attach and then it will cause the protein to change shape and it's released on the other side. Now, there's a very important channel protein called aquaporin that greatly increases the rate of passage of water molecules. Aquaporins are composed of 4 polypeptide subunits, and they each form a channel for the passage of water. Listen to this statistic: over 3 billion water molecules can pass through per second in an aquaporin. And so in this diagram, this is actually a diagram, the 3D model of an aquaporin, where the yellow balls here are the heads of the phospholipids, the green is the phospholipid tails, and then the red and gray are the water molecules on both sides of the plasma membrane. The aquaporin is the blue alpha helices that exist here through the membrane. So water molecules will travel through the aquaporin to get to the other side of the membrane. 3 billion water molecules per second can travel through an aquaporin. It's just fascinating. It's just amazing. So the selective permeability of a membrane is dependent on both the lipid bilayer and the specific transport proteins it contains. And this is all fueled by diffusion. So in the next video, we're going to go through passive transport and review diffusion.